In this video, we will discuss about a host defense system in the bacteria, and that not only targets the DNA but also has a second line of defense. It will also target some of the RNA, foreign RNA transcripts. So, and we are already familiar with the DNA part of this bacterial immune defense system, that is popularly known as CRISPR system, where what though I have already covered this CRISPR part in detail in a series of videos. You can go and refer them just for a brief information or just for a revision. That is what it CRISPR does is that whenever a foreign genome enters the cell, this CRISPR will detect those foreign genome based on the PEM sequence and add this foreign genome into its own DNA sequence, and this thing is done by different kind of CRISPR, different kind of this Cas protein that is categorized into different types such as type 1, type 2, type 4, type 5. However, with time, virus has evolved and tried to evade this kind of CRISPR, this kind of first line of immune response of this bacteria. And some of the evading techniques that are used by the viruses are this anti-CRISPR protein and this REC binder, restriction binder inhibitor, or this restriction binder inhibitor, or this uh, regulation disruptor, anti-CRISPR protein. And sometimes what happens, since this CRISPR detects any foreign and targets any foreign antigen at the PAM sequence, sometimes the PAM sequence is mutated in the virus, thereby this CRISPR first line of immune defense will not be able to target this kind of viruses. And the other things that can help the virus prevent itself from this first line of immune defense is this glycosylation, and we have covered glycosylation in many of our videos. You can refer them. So what it will does is it will the proteins the glycosylation act as a physical barrier, and where this a first line of immune defense will not be able to interact with the PAM sequence of the foreign antibodies, and thus the transcription of the viral genome will occur and the viral RNA will get transcribed. So here comes the second line of defense of this host immune system, and that is known as RNA targeting. So in this case, RNA targeting, what it will does is there are two kind of CRISPRs. There are two kind of Cas proteins, CRISPR Cas system uh, that is found efficient on this RNA targeting, and these are type 3 and type 4 CRISPR Cas system found efficient on working and efficient on targeting the foreign RNA transcripts. And however, they do not target the whole RNA transcripts, but they will only target some of part of the RNA transcripts which are commonly called as this lytic transcripts. So this second line of immune defense will not be able to completely eliminate this RNAs. However, they will eliminate only the lytic part of the RNA, and because of this, a new steady state will be generated where the host needs to get host will get equilibrated or tolerate with some part of the viral transcripts that are now been translated using the host machineries. So this is known as the second line of defense, and which has some disadvantages. That is, here some part of viral transcripts get translated, and this is in the region that cleaved up by the Cas9, and this viral RNAs are then been this lytic RNAs in simple terms. This lytic RNA transcripts are then degraded and by this CRISPR Cas system, that is type 3 and type 6, and are then be recycled back that can be used by the host machineries for their for the transcription of other host other host self RNA's sequence. So that's all for this video. Yeah, if you need to understand more about this first line of immunity and second line of immunity of this bacterial immune system, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.